Natalia Gomez-Ospina. I'm a physician scientist at Stanford University. Families with Krabbe disease know that bone marrow transplant is currently the best option they have, but it doesn't work very well. It has to be done early, and the immune matching has to be close. And even with all that, the damage to the central nervous system doesn't always stop. And so we're working on a different approach. Instead of using someone else's cells, we take cells from the patients themselves, and we use CRISPR or genome editing to fix the problem genetically and put the cells back. Uh, because they come from the patient, um, the immune system doesn't reject them. And because these edited cells can naturally travel from the bone marrow to the brain and the nerves, where exactly where Krabbe causes the most damage, to deliver the enzyme there. So that's how they're effective. And the idea is to have a one-time treatment that is safer, more targeted, and available to more patients. So Krabbe disease has been a focus on mine for a long time, long enough that I was already working on it using sort of my own lab startup funds before there was any dedicated grant money, before any dedicated grant money came through. A colleague of mine invited me to speak at the Krabbe Disease Research Network meeting, and it was there that I first heard about the Rosanna Family Foundation's grant program. It felt like a natural fit, and the foundation obviously understands the urgency that comes with this disease, and in a way that even larger funding agencies don't. And meeting that community in person uh, allowed me to make the connections that felt very kind of immediate and, you know, uh, very helpful. So I am a medical geneticist, and which means the patients that I see are all rare patients, all rare patients. Many of the patients that I see in clinic are often children with conditions that don't have treatments yet. You sit across the family who, especially who has just received a diagnosis of Krabbe, and you know what's coming, and you know that the tools that we have are inadequate, and it's hard to walk away from that. And the science of gene editing or CRISPR finally kind of gave us researchers a real tool to try to change that. And Krabbe fits our platform. We already had a genome editing platform that could fit other diseases, and it fits it especially well because it's a single gene problem, and we could use stem cells to try to improve it. Um, the fact that it's working in the lab, we have shown in animal models that gene edited cells migrate to the brain, that they become functional there. They're able to deliver that missing protein. Um, and that's not obvious. For a long time, people weren't sure that this strategy would work that well in the brain. Uh, we have also done a lot of work on our genome editing platform, initially developed it for other conditions. We know that it works well across multiple conditions. We know that it's safe, which is the most important thing. Um, and so the biology is on our side. And the question now is translating it into treatment for families they can actually access, which is a much more complex problem. Um, the best part of my job is being a physician scientist, the best and the worst. The best part is being a physician scientist and being in the clinic with families and then going back to the lab to work on their issues. Like I have very clear vision of who I'm helping. Uh, you know, we're not curing cancer or, degeneration, but I can see those families. So those worlds push each other forward, and Krabbe particularly is very motivating. because of their families and the extreme need. The hardest part is the pace. Rare diseases can move very fast in children, and the science doesn't always move as fast as families need to. Funding is a constant challenge uh, for diseases that affect, fewer people, get less attention, um, which is exactly why organizations like the Resident Family Research Foundation matter so much. Um, I wish I had learned to fail more gracefully. In science, failure isn't the opposite of progress. It is progress, and every experiment that doesn't work can tell you something true if you did a good experiment. And every grant that gets rejected, if after you get past the pain, pushes you to sharpen your thinking and your writing. But early in your career, failure feels very, very personal. I think I'm still sort of in that stage. And you spend a lot of energy trying to avoid it or recover from it. So I've seen some researchers that are very successful and they move the needle fastest. Those who move the needle the fastest are the ones that, take risks. So getting to the clinic if you can, if you're a basic scientist, understanding what a family is actually living through changes how you prioritize, how you organize your research questions, and it just gives meaning to your work. If you can lose that when you're in the lab troubleshooting, little things constantly. So getting to, or find the patient's community early, ask your PIs to bring you to a family meeting if you're working on rare diseases. Um, Families living with Krabbe and in general other lysosomal storage diseases are some of the most, they're very engaged scientifically and you can learn from them. And also I would say that the field of genome editing is young enough that trainees can make a genuinely foundational contributions right now and a real scientific frontier. So um, I have two small kids. Honestly, they keep me pretty busy just on their own. Uh, Physical activity is really important to me. Swim, I bike, I lift weights, my age, which I'm not gonna say what it is. You got to stay active, constantly switching things up because, my body appreciates the variety. Um, Outside of, chasing kids around and trying to stay healthy, the thing that fills whatever quiet moments I have is listening to audiobooks. I go through, one or two a week, and I don't do it to learn, it's not something so laudable. I really do it to try to read fun things or slow reads that can help me keep my mind engaged, but also prevent me from perseverating on problems or just leaving the lab behind or the clinic. I recommend it as a great strategy to for a mind that tends to persevering on things is a good way of letting go, tricking your mind and thinking about something else.